Lipids pose actually several interesting biophysical problems in terms of digestion and absorption, and that's because lipids, by definition, are insoluble. That means we have two challenges. We need to break the lipids down into an absorbable form, but we also need to make sure that they're soluble and can pass through our gastrointestinal tract. Most enzymes work in aqueous environments, so for them to work effectively, we need to have a solubilization step in addition to the regular enzymology that we've been talking about before. In this video, we're going to talk about the three important lipases that break down triglycerides: lingual lipase, gastric lipase, and pancreatic lipase. We're going to talk about what the role bile salts play, specifically to fat absorption. We're going to talk about the transport mechanisms by which lipids get across membranes and how they differ between different lipid classes. We're going to talk about the process that happens in the enterocyte where lipids are repackaged for transport as chylomicrons for delivery to the rest of the body. The major component of dietary lipids is in the form of triglycerides. About 95% of the lipids we eat are in the form of triglycerides. The average man consumes about 95 grams per day of triglyceride, whereas the average woman consumes about 65 grams. Triglycerides are the main source of the essential amino acids, the omega-3 and omega-6 polyunsaturated fatty acids. We also consume about 300 milligrams per day of cholesterol, generally from animal products, and 1 or 2 grams per day of phospholipids. Our digestive system therefore has to have pathways to break down triglycerides, cholesterol, and phospholipids to put them into an absorbable form. Remember, lipids by definition are hydrophobic molecules. That means they tend to group together in large globules. But we need to prevent that. We need to emulsify the fat into smaller globules. We do this through two things. One is by coating it with a soap-like substance called bile acids. The second way we do it is using mechanical forces such as churning and passing through sphincters to help break large globules down into smaller globules. Eventually, these smaller fat globules are broken down into even smaller things called micelles. These are the absorbable units for lipids. The first step of digestion occurs in the mouth. The first enzyme is called lingual lipase. It's secreted by glands underneath our tongue, and it works best actually at low pH. That means that while it's made in your mouth, it doesn't start working until the low pH environment in our stomach. When lingual lipase is active, it has a preference towards medium-chain fatty acids at the SN3 position. That means the number 3 carbon of glycerol. It tends to therefore generate diglycerides and free fatty acids. In addition to lingual lipase, within the stomach, gastric lipase is also secreted. It's secreted by the chief cells of the stomach. It has a different preference. It prefers both short and medium-chain fatty acids, but prefers to cut at the SN1 and SN3 position. It works mostly in the stomach, but continues to work a little bit in the small intestine as the stomach acids become neutralized. Also happening in the stomach is the start of this emulsification process. The strong muscles of your stomach and the churning of the liquids in the stomach help to break down and coat these lipid particles. Another important step is wear and tear of these flat globules. As they squeeze through the sphincter muscles, especially the pyloric sphincter, the result is breaking down of lipids into the smaller globules. The third and most important enzyme in triglyceride digestion is called pancreatic lipase. It's made in the pancreas and secreted into the small intestine. It has specificity and preference towards long-chain fatty acids on both the SN1 and SN3 position. Again, generating mostly monoacylglycerols with the fatty acid still on the SN2 position. As the gastric lipase, lingual lipase, and pancreatic lipase function, more and more triglycerides are broken down into more monoacylglycerol and free fatty acids. These can then become packaged into smaller and smaller droplets, eventually emulsified droplets called micelles. Formation of micelles is facilitated by coat particles called bile salts. These work as soaps. They cover the surface of the micelle and allow for transport into the body. Importantly, bile salts are made from cholesterol. In fact, this is the major way by which we get cholesterol out of our bodies, is by converting it into bile salts and then secreting it into our gastrointestinal tract. Bile salts are initially made in the liver, stored in the gallbladder, and released into the small intestine as they're needed. The signal to release bile salts is driven by CCK. The recycled bile salts, and most of our bile salts, about 95% of them, are recoverable, can keep being reused. The hormone CCK is released from enteroendocrine cells called eye cells in the small intestine. When it comes in contact with food, CCK is stimulated to be released, it signals to the pancreas to release various lipases, and it signals to the gallbladder to release bile, and signals to the stomach to prevent more food from going into the small intestine, until the food that's currently in the small intestine can be efficiently digested and absorbed. Bile salts come in a large number of varieties. They're all synthesized from cholesterol. The first step, which occurs in the liver, is conversion of cholesterol into either cholic acid or N-deoxycholic acid. These are known as primary bile acids. Once they're released into our gastrointestinal tract, they may come in contact with gut bacteria. In that case, they can be dehydroxylated into deoxycholic acid or lithocholic acid. All four of these precursors, the primary bile acids and the secondary bile acids, can also be conjugated when they transport back into the liver. Since bile acids can be recirculated many, many times, all of these different precursors can have either the amino acid glycine or the amino acid taurine. Taurine's an amino acid that we don't use for proteins. Those can be attached to any of those four amino acids, meaning that we can generate 8 different bile acids. These are called conjugated bile acids, once an amino acid is present. Each of these different bile acids can be present at different levels, depending on the activity of our gut microbiome and the efficiency of conjugation. They also have different biophysical properties. They can coat lipids differently, they can be reabsorbed at different rates, and they can even signal differently throughout our bodies. The first step of bile acid synthesis, those that make the primary bile acids, is shown on the left. The rate-limiting enzyme is called cholesterol 7-alpha hydroxylase. It's an important step by which we convert cholesterol into a primary bile acid. The bile acids all have a similar function. They will take a fat globule that is broken down and coat the smaller lipid globule. These emulsified droplets, which contain a small number of lipids but are coated by the bile salts, are very small and are actually the form that can be absorbed. Since bile salts are amphipathic, like phospholipids, they are useful for coating neutral lipid droplets. Let's return to the other two lipids, cholesterol and phospholipids. Most of the cholesterol we consume is in esterified form, and esterified cholesterol is not well absorbed by our bodies. That means, in order for us to absorb cholesterol, the first step is we have to cleave off the fatty acid from esterified cholesterol. That free cholesterol and the free fatty acid can both be absorbed. The enzyme sterol esterase is made in the pancreas and secreted into the small intestine. The last lipid class is phospholipids. We eat about 1 or 2 grams per day of phospholipids. In order to convert phospholipids into an absorbable form, we need an enzyme called phospholipase A2. This enzyme cuts off a fatty acid from the SN2 position of a phospholipid. The resulting lipid, which now has a fatty acid on the SN3 position and a phospho head group on the SN1 position, is called a lysophospholipid. Lysophospholipids and free fatty acids are absorbable into our bodies. Throughout all of this digestion, emulsification, and coating with bile salts, we're going to end up with free fatty acids, mostly monoacylglycerol with fatty acids at the SN2 position, but maybe some diacylglycerols, free cholesterol, and phospholipids in the form of lysophospholipids. These all can combine into a micelle which can be coated by a bile salt. We actually don't know a lot exactly about how these lipids are transported into the enterocyte. Some people believe that there are actual transporters on the surface of the enterocyte that facilitate this diffusion. Other people argue it's mostly passive diffusion from the lumen of the gut into the enterocyte. But either way, the lipid contents get into the enterocyte, whereas the bile salts, which are amphipathic, cannot cross that membrane barrier. The bile salts are then free to be recirculated and used over and over. Now, what happens to the lipids once the lipids get into the enterocyte? Well, it depends on the actual length of the fatty acid. Let's start with shorter chain and medium chain fatty acids. Those are fatty acids with shorter than 12 carbons. Because they have shorter carbon chain lengths, tend to be quite soluble. Therefore, they can diffuse into the enterocyte and then diffuse into the blood. There, they can be transported by a protein called albumin and can move to other tissues directly. However, most fatty acids we consume are long-chain fatty acids of more than 12 carbons. In those cases, they need to be reesterified back into triglycerides. Cholesterol needs to be reesterified into a cholesterol ester. These hydrophobic neutral lipids are then packaged into apolipoprotein particles called chylomicrons. Chylomicrons then serve as transport vehicles to take lipids from the enterocyte to the rest of the body. Shown here on the right is a schematic of what a chylomicron, or a general lipoprotein particle, might look like. The neutral lipids, triglycerides and cholesterol esters, are in the interior. The coat surface layer is phospholipids, and there are proteins embedded on the surface called apolipoproteins. A chylomicron is important for the transport of reesterified lipids out of the small intestine, into the lymph, and to the rest of our body. These are essential for solubilizing these insoluble triglycerides and cholesterol esters. Once generated in the small intestine, those chylomicrons transport into the lymph and then move on into other tissues, mainly muscle and adipose tissue. There, using the enzyme lipoprotein lipase, muscle and adipose tissue can take the fatty acids out of the chylomicron particle and store them for either storage or use, depending on the tissue. Once a chylomicron has most of its triglycerides depleted, it's known as a chylomicron remnant. It contains the phospholipids, the apolipoproteins, and some cholesterol. This can then be reabsorbed as a remnant into the liver, where those phospholipids and proteins can be recycled if needed. In summary, lipid digestion and transport requires two things to happen simultaneously. You need enzymes to break the lipids down into smaller, more soluble lipids, and you need mechanical forces and bile salts to help emulsify the insoluble lipids into soluble factors. The end product are micelles. These are made up of partially digested lipids and bile salts. This aids in the absorption of those lipids across the border into the enterocyte. What happens next depends on the length of the fatty acid. Long-chain fatty acids are reesterified and transported as chylomicrons. Medium and short-chain fatty acids are directly transported into the blood and go to the liver. The chylomicrons are made from apolipoproteins and phospholipids. This helps to solubilize neutral lipids and allows for them to be transported to other tissues. After transport and delivery of lipids to other tissues, the chylomicron remnant, which is cholesterol, phospholipid, and protein-rich, can be absorbed in the liver and reused if needed. Lipids are a major energy source as our diet, but lipids also provide several different challenges in terms of absorption and digestion, mostly because lipids are by definition not soluble. Therefore, we require the concerted effort of enzymes, bile salts, and mechanical forces to break lipids down into a form that can be effectively absorbed and transported both into the enterocyte and then to the rest of our bodies for use and for storage.